So thank you for joining us for our cafe this morning or this afternoon. Obsessed with observations, become an iNaturalist super user. And uh, we'll- but as I said, this is registered as well as added to our website. And if you have ideas for future cafes, please let us know. With that, I will turn it over to Nicole Machuca from the Field Museum.
Hi everyone. First, is that Laura's internet or my internet? I don't know, Jing-Yi, were you also hearing? I'm worried it's my internet.
I think it was Laura's
Okay.
internet, so
Okay.
it should be fine.
Okay,
Well,
sorry.
welcome everyone. No, again just worried if it was me that I would be breaking up. So yeah, hi everyone. I am Nicole Machuca. I am an environmental social scientist at the Field Museum. Talk a little bit more about why I am presenting on this today, but first want to make sure that folks see in the chat that you are welcome to add where you are joining from today. And if you do like to snap pictures of critters, please list those in the chat as well. But Will, Jing-Yi, and I are here today to talk not only about iNaturalist and how to become obsessed as we are with making observations in iNaturalist, but we want to highlight that this is all part of the 2026 Chicagoland City Nature Challenge. So if you haven't yet joined on the app or on your browser, please do that while we're going over our presentation today. But yeah, like I said, I'm Nicole Machuca, she, her, environmental social scientist. I am a hobbyist naturalist, I suppose, jack of all trades, master of none. We've included all of our iNat handles on here if you want to follow us on iNat. And I love to take pictures of bees, pollinating flies, and pollinating wasps. And is Will here? Pass it to Jing-Yi, maybe?
Okay, so my name's Jing-Yi. I'm currently a research associate at the Field Museum. I'm trained botanist, but also interested in like plant-pollinator interaction and how those affects plant evolution. So I do a lot of like iNaturalist photos for plants, birds, and flies, and all kinds of organisms.
Great. And then if Will comes, when he comes on, I'm sure we'll have him introduce himself. But just to give you a little bit of an overview of what we're going to talk about today, hopefully very quickly, iNaturalist 101, in case we have some folks who are new to the iNaturalist app and world and need some of those basics. Will, Jing-Yi, and I are going to go over observation tips of the critters and kingdoms that we're obsessed with, some of our lessons learned. And we'll talk a little bit about identification and how to really bump up your iNatural skills by going over the advanced features. And then like someone put in the chat about how to become part of the challenge. I'll talk about that at the end as well. So iNaturalist 101, what is it? It is both a website and an app from your phone. All of my screenshots, you get to see the time that I took the photo. Don't judge too hard. But if you create an account, it is free and it will link either in your phone and your browser. So here are two different views from my web browser and my phone. And I'll talk more about that in a minute. But it's basically a community science application where anyone across the globe can submit a recording or image of a critter that they've seen. And there are now two versions. Again, if you're new, maybe this is old news or new news or something you wanted answered, but there are two versions of the app. I use an iPhone, by the way, so this is what my app store on the left looks like. And I use iNat Classic, but you can see in the center two different views of iNat Classic next to iNat New app. Same organisms, same data, just the view is different, and there are pros and cons to all three: web browser, iNat Classic, and new iNat app, which does have some features that Classic doesn't have. I still like using Classic because change is hard. So no difference. You can use whichever app you want to use. And just we wanted to talk a little bit because all three of us are going to have images of things close up. So wanted to talk about you can be a phone user and become a super observer like me. Or you can use a camera with a macro lens and become a super observer like Jing-Yi and Will. So just want to point out on the left, you'll see two photos from my phone where I have an iPhone 10. It's an old iPhone at this point. I still am able to make meaningful documentation or observations. I do use a lens attachment on my phone. And I'll talk more about that. And then there are, if you are a camera user, but you want to take macro photos or macro lenses and classes around the region and globe, Bugshot has been referred to me if you want to really up your macro photography skills. So how to submit an observation. Because I'm a phone user, I'm going to show you from my perspective. But first you would click, again, my view would have all the way in the left, you see the observe button on the bottom. And then it will pop up a camera, photo library, or record sound option. I do record sound because I do like to record the common nighthawk. I live on the southeast side of the city. And the Field Museum, we also have a couple common nighthawks. So I do record sound every now and then. You can take photos directly from iNat app's camera, but I do a lot of my uploading from my photo library. And then what we really wanted to highlight for you all today is this next middle image about what did you see and your location. So for all of my photos, I give my phone permission for it to record the metadata. So location populates automatically for me when I'm uploading photos. But if you do not have your phone set up that way, or if you're uploading to the browser, for example, you would have to manually go in and edit location, which is the image all the way to the right. And then again, what Will, Jing-Yi, and I really wanted to highlight for you today is please always put something in what did you see box. And Will is going to talk a little bit more about that later. And this is a view of the browser version of the iNat. And one of the pros of uploading that way is that you can upload a bunch of images at one time, and then you can combine the species together. Or if you know, like I have some margin beetles, leatherwing beetles on the right, you can, it'll combine them so you won't have to each time put species name, location, notes, that sort of thing. And another way you can, if you're new and you want to bump up your iNaturalist contribution, you can add some annotations. Like if you find a dead organism, you can mark that. If you also find evidence of the organism, like it's an egg or it's the molt of some sort of organism, then you can leave that as well. And then you can have a lot of fun on the browser by searching different, I like to just find out what's around because Will is kind of our fungi guy in this webinar today. I was curious how many species are in Chicago. So according to what has been submitted in iNaturalist, we have over 1,000 species. So I like to search stuff like that. If you look on the right and the top, here's an example of another way I'll play with the browser is I'll look at a species and I'm like, oh, narrow-headed marshfly. Okay, I'm curious about this family of hoverflies. Let me learn more. And then I can view all, and then I can select Chicago again because I am a Chicagoan. I am curious, and I can see that we have 68 species of hoverflies in Chicago. So a lot of things you can do to play with the app behind the scenes. And with that, I believe I'm passing it to Will. Are you here, Will?
Yep,
Yeah.
right here. You guys hear me okay?
Yes, and you want to introduce yourself as well?
That sounds good. Yeah, sorry, I missed the intros at the beginning. I am not a Microsoft Teams person, and there was some some troubleshooting there at the beginning. But happy to be with you all now. My name is Will Yaegar. I'm a first-year graduate student in the Department of Ecology and Evolution at the University of Chicago, and I study rapid adaptation in plants. Um, but in my free time, I'm also a very passionate amateur botanist and mycologist, and I've done research related to plants and fungi before this. And so most examples that I'm going to talk about today are going to relate to fungi. There'll be some plant ones as well. But I guess before we get into any like kingdom specific stuff, we're just going to take you through some general observation tips. And cool, perfect. I was going to ask Nicole if you wanted to keep sharing the screen and just scroll through that. That works great. Thanks for doing that. Um, so I must confess that I am occasionally guilty of making bad iNaturalist observations. And when I was making this presentation, I was looking through my recent observations. I found this one from a few weeks ago when I was in Arizona. And I guess just to start, like if we're looking at this photo right here, it's really not clear what we're looking at all, right? This is kind of just a photo of like a landscape. It looks like there are various plants there, but it's blurry. It's taken from far away, and it's not clear like which of the many plants in the photo I actually meant to observe. So this is like a great example of what not to do. And just like for some contrast, on the next page here, on the next slide, you can see an observation of the same species. So this is Listhenia gracilis, common goldfields, same exact species that I attempted to observe in that first one, but a much, much better observation. And why is this a good observation? So it's a very sharp photo, right? It's zoomed in. We can actually see like some diagnostic features of the plant that we're trying to identify, like the petals and the sepals and various parts of the interior of the flower. You can also notice if you look at the bottom of like the thumbnails here on this observation, there are three different photos, right? And so on the next slide, we're going to see what some of those other photos look like. If we could switch to that. So you can see this person not only took a great, initial photo that was close up and zoomed in, they also got photos from multiple angles. So this other one here shows like the top of the flower, where you can really see like the number of petals, which is often important in identifying flowering plants. This last photo I really love too because it shows like where it was growing. It shows the habitat, it shows other species that were growing nearby, and it includes this person's hand for scale. Sometimes if there's no like object in the photo for scale, it can be really difficult for an identifier to know like how big the thing in the photo is that they're looking at. So this was like a helpful thing that they did. So this is like, what a better observation than mine looks like. And what a good observation looks like, though, is also going to vary a little bit from group to group. So here's some more general principles, and then we're going to go into like the specifics for different types of organisms. One good principle, with a few exceptions, is that like in general, you want to be photographing one organism per observation. Okay, so like having just one flower right in that last photo was a good thing, because it could be the case that you have multiple similar looking species growing near each other, or like if you take a very zoomed out photo and you only have one or two photos, it's often just not clear. Like if you're in an area that has a lot of different species occurring side by side, it's just not clear sometimes like which one you meant to identify with your particular photo. So like one organism and like specifically one individual per observation is a good policy to follow. I think Nicole mentioned this earlier, but you really want to make sure that if you're photographing on a phone or like a camera that has a GPS built into it, make sure that everything is set up and turned on so that the location is going to be recorded in your observation, because observations, no matter how good the photos are not going to be super useful if we don't know where they were occurring. And then sort of like I was saying in, the last example there, the common goldfields, you really want to take multiple photos at different angles and distances and try to show all of the visible parts of the organism that you can. Sometimes if it's something that's far away or in motion, like a bird or something, that'll be challenging. But like really, this is just about providing as much information as you can in your observation to help identifiers. And part of that too is, getting a sharp in focus image. You can include some objects for scale. The example I have on the right here is some of you guys might be familiar with Joey Santor, who's a botanist from the Chicago region, who runs a YouTube channel called Crime Pays, but Botany Doesn't. And he has a tattoo on his finger with a centimeter ruler that he often includes in his photos for scale. I don't think we'd expect that level of dedication from all of you guys necessarily, but even just a finger without a tattoo is useful because it kind of gives an approximate sense of size for the thing that you're holding up. And then finally, if the observation that you're making is of like a captive or a cultivated organism, so this could be a pet, this could be something that's in your garden, this could be something that was like planted in an urban park, you want to mark that. There's like a little checkbox that you can check that says captive/cultivated. And for the City Nature Challenge in particular, we really want to try to avoid making these kinds of observations because we're really interested in understanding like the biodiversity that's present in our city that's like not been explicitly put there. Sometimes it's not going to be clear whether something's captive or cultivated, and those kinds of situations are okay to upload. But like if it's like a bush or something in a park, like a hedge, that's very clearly been put there. You can still observe it if maybe you're curious about hedges and what kinds of plants people are using for hedges these days. You can still definitely make observations of those, but just please make sure that you're clearly marking it as this is a captive/cultivated observation, so that it doesn't end up like polluting the databases of like research grade quality observations that scientists might be using for range maps and things like that of species that they're assuming are being observed in their native range, or maybe in an introduced range, but like accidental as opposed to deliberate introductions. So anyway, those are some general tips. So I mentioned that I have done a lot of work with fungi in the past, and so here I have like an example of what a good fungus observation could look like. This is Trichospora thelephra. This is a fungus that I found growing in the jungle in Peru about a year ago. And so I mentioned on the previous slide that generally we want to include like one individual per observation. With fungi, that is less true. With fungi are like super variable in terms of their appearance as they go through different developmental stages, and they often occur like in groups. So, if you find a fungus, that's going to be like one instance in which you would actually want to include, maybe multiple individuals in a photo, or, photos of multiple different individuals that were very close to each other in an observation. But again, it's like you've got to be confident that it's actually the same species, because we don't want multi-species observations. And I guess something to note about fungi in particular is that, and I'll show more examples of this on the next slide, but with fungi, I see a lot of fungal observations on iNaturalist where it's really difficult to tell what it is, because people just have like one photo of the top of the fungus. And the top of the fungus is usually not enough to identify it. We really need to see the underside, which in mycological terms is called the hymenium, and this is like the surface on which the spores are being produced, right? So if you think of like a normal grocery store button mushroom or portobello mushroom that you might grill up, that underside is going to have gills, right, that are producing spores. For other species like this one, there's going to be like tiny pores or teeth. I don't know if you can see on the right there. I guess I kind of made it a bit small, but that pink surface that you can see is actually covered in these tiny like little teeth, like little spikes, and spores are being produced on those spikes. And so like knowing what the underside looks like is going to be super important for allowing people to help you identify it to the level of genus or species. And the same thing goes for looking at the stem of the fungus. So we don't just want to look at the cap, from above. We don't just want to look at the underside of the cap. We also need to see the stem, and specifically like the base of the stem, because often there can be features about the base of the stem that allow us to more confidently assign it to a genus or a species. Now, in order to see the underside of the fungus or the base of the stem, you might have to pick it, right? And from like an ecological standpoint, this is generally fine. And the reason for that is that mushrooms are like fruiting bodies of a fungus. So the mushroom is not the whole organism. It would be like if you pick a mushroom, it's kind of like picking an apple off an apple tree, right? This is like a structure that the organism is producing in order to be able to reproduce and spread its spores in the case of the mushroom, seeds in the case of the apple tree. But there's a whole rest of the body that, you know, in the case of fungi is going to be either underground or like in the rotting log or whatever that it's growing from. That's going to be totally fine and is going to keep living there and, you know, be able to survive this particular encounter. So like for observational purposes, like it's fine to pick a mushroom. That said, you know, if there's a bunch of them, like you probably only need to pick one or two, right? And you know, there are certain places where this kind of thing is discouraged because they're visited by lots of members of the public. So the forest preserves of Cook County, for example, don't allow mushrooms to be picked, whether it's for, you know, eating or for you know, science, unless you've got a permit, of course. And so another alternative that you can start to think about in scenarios like this is well, one, you could try to like use your selfie camera on the front of your phone and like stick it underneath the fungus and try to get a picture of the underside, you know, without actually disturbing it from its natural habitat. Or the other thing that some people do that I think is kind of neat is you can carry a little mirror, like a little makeup mirror, and you can put that on the ground underneath the fungus and then take a picture of the mirror. So those are like some creative solutions to how to, you know, leave the fungi undisturbed and still get the information that we need for identifying them. Let's see. I think at this point I'm turning it over to Jing-Yi then, who's going to talk about plants.
Yep. So I think for plants, the same general rules about like good observation also applies. For a lot of plant observation, we see like people tend to be really just focused on things get them attracted to. So you got a lot of photos of only having certain features of plants, like just flowers, or just take a random photos out of the plants they found. But for plants, you also want to have a really comprehensive observations to see all the characters that helpful for identification. So ideally, you want an
Sorry,
overview, some
Jing-Yi,
close-ups.
just ask people to mute. Yeah, somebody's unmuted. Sorry.
Okay, so we want to have some overviews, some close-ups, some also underside, like Will mentioned, also apply for plants. And if ever got chance, also could also observe some microstructures on plants. So I'm gonna use this American groundnut from Indiana Dunes State Park I observed a few years ago as an example. So you might be attracted by the flowers, like the maroon structures in the pictures you see. So you can take an overview of these plants. It helps us to understand how these plants grow and the habitats it lives in. And then you can move closer to the plants and try to see how the plants actually grow, to see how these flowers actually attach to different parts of plants. For example, for this groundnut, it's a vine. So a lot of times these vines just grow tangle with other plants. So it's helpful to actually separate the right leaves that attach to the flowers. So after finding the right leaves, you want to take some close-up photos of the leaves showing on the right-hand side. And a lot of time people just forgetting about, it's also important to flip the leaves around to see the underside of the leaves. So next slides, please. Yeah, and then you can move to other structure of the plants to take another close-up photos. For example, here is the flowers of the plant, the groundnuts. And it would be helpful if you notice any interesting structure on the plants. You can take some, if you have suitable like tools, you can take some close-up views of the microstructures. I will talk more about this later. So what are the features to look for plants in general? Next slide. Yeah, so for plants are a diverse group, so there's a lot of things to cover. But in general, you want to look at leaves, sometimes barks if it's a tree, and flowers and fruits, and spores if it's a fern or other related non-flowering plants. So for leaves, you want to have a photo that kind of cover the general features that maybe you can tell the shape and sizes of the leaves, and you can clearly see the borders of leaves, whether they are entire, they are lobe or not. And for example, this groundnuts, this picture is actually showing full leaves. But even you don't know about it, you can still take a photos of the entire structure to show that how this smaller leaflets actually arrange on the plants. So leaf arrangements actually helps a lot of, for plant identification. And if possible, also take a closer view of the leaves that you can show the patterns of vines, which called venation, that also help in identification for a lot of cases. And the microstructure I mentioned in the previous slides, you can see there's some hairs growing on the underside of the leaves. So if you can also notice those structures, sometimes it will also help with the plant identification, or just make the observation better with more characters. And for flowers, you also want to have an overview of shapes and sizes, also arrangement, how this flower is growing on the inflorescence. You want to have pictures of showing the front view of flowers, but also the side views of flowers. And sometimes it's showing in the lower pictures here, inflorescence with flowers not opening yet could help a lot because they show the underside, the other side of the flowers that cannot be viewed in the opening flowers. And you can see the arrangement way better than the up photos in the full blooming flowers. Next slides. So at different times of the years or some different plants with different phenology, you might also encounter fruits. So same general rules apply. You want to see how this fruit's growing on the plants. You want to see the colors, the shapes, and it will be also helpful to notice how the trunks or the twigs growing and the texture of those. And for non-flowering plants, for example, especially in ferns, you want to see if you can see spores. A lot of times, spores growing on the leaf underside. So you also always want to flip the leaf of ferns around to see, for example, this wood fern, the spores are growing on the underside of leaves. You want to see how they grow, the patterns, and also the morphology of these spores. And for some groups of ferns, the spores will grow on different types of leaves. For example, these are called the fertile fronds. It might look like a different plants, but if you look close enough, you will find these leaves growing together from the same plants. So these are like a kind of different types of leaves that only just growing for reproduction in these ferns. So yeah, so there's a lot of like plants have a great diversity. So even for me, for a lot of time, I will find myself not observing the right character to identify. But plants have the benefits of being seasonal, like they are not moving around. So you can they have this benefits for you to always go back to the same plants to revisit them and learn more. So once there's someone coming to say, I want to see these features a little bit more, or you find some resource telling you you need to see like certain leaf structures or like hairs to actually identify this species, you can always revisit them at the same location, try to learn more about certain species, certain individual, certain plants. And it's always important, as Will mentioned, especially for City Nature Challenge around Chicago, you will see for a lot of time plants are being cultivated, especially in flowering beds. So you can still upload those onto iNaturalist, as Will mentioned, but just need to mark them as cultivated. And it might sometimes be really helpful if you're doing association with other organisms, which I'm gonna talk about next. So to extend from just plants, you can always look for other diverse organisms that kind of live on plants or alternating their structures. So what are the things you can look for? For example, I'm showing just some like examples for really diverse groups of organisms lives on plants. You can look for goals. This could be arthropods or that kind of change induce like plant abnormal growth. So if you see really strange structures, sometimes these are goals induced by these different organisms. There's also special kinds of insects that kind of live inside the leaves that creating these patterns are leaf miners. And there's also fungi that kind of parasitize on plants. Like, for example, the powdery mildews, they're like white powders you see on plants. They're actually fungi growing on plants. And there's also rust fungi showing also here that kind of like a orange structures you'll see like really abnormal for leaves you see. And there's also more familiar microorganisms like insect larvae or pollinating insects or birds that could associate with plants. So not only plants, you can also extend your observation for more diverse organisms. So next slide. Yeah. So where to look for this different things? Some of this fungi, they will grow on the stems of trees like this black nuts that's actually pathogenic fungi. And a lot of galls actually grow on the different sides of leaves. So sometimes they grow on both sides of leaves, sometimes only on the undersides. And sometimes rust fungi will also grow on flowers and fruits. So there's all kinds of possibility you can look around for different things that grow on plants. Also showing here is the gold midges that change the structure of plant stems. And this leaf mites actually growing on leaves of poison ivies. So there's all kind of possibility you can look around for different things. So next slide. And for this different galls, leaf miners, rust fungi, powdery mildews, for a lot of time, they are really host specific. So having the knowledge of their host plants are really important. And ideally, you, after finding some of this, like for example, I having this gold wasp growing on burr oak here. Finding this goal, after finding this goal, you want to have a more in focus photos of this, but you also want to have an overview photos of how this structure growing on plants. For example, I take another photos of this burr oak, so you can understanding what is the whole species. And then you can use the observation fields that in iNaturalist interface that there's two specific observation fields that people use more frequently for goals and parasitizing fungi, the host plants and host plants ID. So for example, you can see for this observation, I have annotate for the host plant ID and host, I have annotate burr oak as the host plants for this. And there's another interesting feature that I feel like people not using that frequently, but I feel like it's really helpful is another observation field is called similar observation set. You can actually link different observation together. So I'm using this European elder that I found in Jackson Park as an example. I usually use for each iNaturalist observation, you'll get ID for the certain observation. So I usually use the ID for the host plant as an input for this similar observation set. So you can see I already input a number that's actually the ID for this observation into the similar observation set. So when you click on this, you will see the pop-up window is showing on the right-hand side. And when you click on the observations with these fields and value, so next slide, you can see all the observation I already linked through this IDs. And you can see there are 3 different organisms I linked with this European elder. And because this European elder is a cultivated tree, so I already marked it cultivated. As you can see, it's actually a casual observation. So the next slide. So through this observation field, I actually linked 3 different observation together. And 2 of them are things I found related to this European elder. One is a powdery mildew species that grow on both sides of the leaves. And there's even a redpoll that I found like during winter a few years ago that feed on the fallen seeds of European elder. So for birds, there's also a really specific association. Sometimes there are certain bird species will more prefer to feed on certain seeds or flowers of plant species. So next slide. So there's a lot of different structures, different organisms you can find on plants. And there's actually a lot of online resource already available on there. You can look around after finding something strange on plants. For goals, there's a really nice website called Goal Formers. I will highly recommend you to like just refer to it to find what you might find on plants. And for field museum, we also provide quite a lot of different field guides that could provide a kind of overview for you to know what you can find for certain plant species, what kind of common goals you can find around Chicago. And for goals, and I think all the parasite fungi, there's actually a really welcoming community on iNaturalist. So as long as you made observation, a lot of people will jump in and try to help you to identify and improve your observation. And I also think having this understanding, like what Nicole do of like searching through the potential organisms you can find around Chicago could be like a great tool for you to do some preparation and have a knowledge of what you can find around this region. For example, because I know the community working on powdery mildews a lot, so a few friends of mine suggesting there's actually a really unique genus of powdery mildews that only growing on the underside of the leaves. So for a lot of time, people just don't know about their existence. And because of that, I kind of start looking at the underside of a lot of trees in the birch family. And I found this unique species that seldomly being observed. And they have these really beautiful structures, kind of really rewarding for me to finding this species. Yeah. So I think I will move to Nicole for the insects parts.
Yeah, thank you. I think that we're a little late in our run of show, so I'm going to zip through this, but I'm going to be talking about how to up your skills with observing insects. And kind of maybe the theme that you're taking away, hopefully, is that angles are pretty important, but just some quick review. Insects can come in the complete metamorphosis from egg, larva, pupa to adults. And that is maybe different than some other species that don't fall within the insecta is that class that it maybe have a nymph stage in there. So those would still be important to document. Just noting that, you know, all life has, as I think Jing-Yi was noting, life can have different moments that will look different throughout the life cycle. So important to keep your eye out for those changes. In talking about how to up your skills for observing insects, particularly in iNaturalist, I wanted to highlight the bumblebee because I think that particularly through this bee spotter resource, if you don't know bee spotter, it's they don't only have guides, field guides, to identify different bumblebee species in our Midwestern region, but they also are a community science project. They still accept observations. I think they are also on iNaturalist and paying attention to bumblebees and iNaturalist, but they still have a website that you can submit observations to. But again, just want to focus because, you know, you might not know that insects can actually be really hard to identify. Maybe that's not surprising or as surprising to hear like fungi and plants. We need different angles. So I have two images here, one of the black and gold bumblebee and one of the American bumblebee. I am not using Latin here. Sorry, folks. But yeah, to someone who's maybe a novice, those bees might look similar. But if someone is looking at the head section of the body, they might be looking at the eyes, how big they are, how large the face is, how slender the face is, how slender the eye is, if there is hair at the back of the head. So these are all, this is one angle to consider taking a picture of, if you can. I'll talk about that in a minute as well, of insects. And then you might also want to catch, if you can, again, it's a lot harder. They're not sessile. They have their own mind. They are moving around, and some of them know you're there and don't like you. So it can be hard to take pictures of insects. But not only to take pictures of, like you see on the right, the top of the thorax, but also the underwing can be important, or this kind of side view can also be important. These are all, so as you see on the left, the northern bumblebee and the American bumblebee. Again, someone who might not know, they might look similar, right? Like look at all those yellow segments on the abdomen. But there are different features of insects that help the experts identify what species they are. And then, like I said, the abdomen. So the different, so they have different segments that might have different patterns or markings. In this case, with the bumblebees, of course, we can see that there are different coloration within their segments, but they're not always as standard as you think they would be. So our most common eastern bumblebee actually has quite a lot of variation in the coloring on its abdomen. So I wanted to highlight, it's a little blurry, sorry about that, but if you Google Nalon's journal, if you look in the red box, Nalon's journal, color variation in bees, that is an iNaturalist user who created this great bee blog that has a lot of different color variation. So again, if you know your bumblebees, that two-spotted bumblebee, there is an unusual color pattern for that species there. So yeah, other angles to capture again, if you can. I love flies and moths, so I wanted to make sure I included those today. They don't get enough love in iNaturalist and just in the urban environment. But yeah, trying to get the side views so you can maybe see the leg coloration again, like wings, different things like that will help those folks identify them. Again, the top side view, especially with flies and other insects, the antenna can be very important. And then we can also try for those insect experts to try to get the front of the face. Again, very hard. A lot of these creatures are wily, flies particularly. They know that maybe it's just me. Jing-Yi also takes a lot of fly and wasp pictures as well. But yeah, the flies tend to know that you're there, and it can be very difficult. But again, for those who are late, I've taken all these pictures on my iPhone, and I have to be an inch away for my macro lens attachment to work. So it is possible. It just means you might not capture some of these angles like if you had the camera like Jing-Yi, because the animals are aware that you're nearby and they might fly away. again, I hope maybe another theme that you're walking away with is there's a lot of diversity. It's not just a mushroom, or it's not just a plant, and it's not just a gnat. So we actually have quite a bit of flying insects that are very, very, very small, that are flying around a flower, especially that you just, again, it just looks like a gnat. But if you look very closely, you'll see that there's a lot of insect diversity, especially around white or yellow flowers if you're looking for flies or wasps. And then I believe this is, ooh, I forget which plant that is. But yeah, pink flowers for bees is the color association there. And my last tip is someone who, again, takes pictures on my iPhone, an inch away from them. I'm not telling you if you are afraid of insects to, try to battle that fear, but just know that things that might look scary are probably like very often stingless. There are a lot of bee mimics out there. And we do have a lot of bee and wasp diversity in Chicago and Illinois and Indiana. And a lot of those species, depending on if it's male or female, might be stingless. And also solitary bees and wasps tend to not care about you so much. And I have been around quite a few nesting colony bees and wasps that when they're feeding on the flower, they don't care about me. They just they're kind of like cats. I feel like that they will communicate when they're unhappy that you're nearby. They kind of like fly right in front of me and look at me. So yeah, I try to respect them. They are wild animals. I'm sure one day I will get stung, but in the 3 years of putting my hand in right an inch from their bodies, I have not been stung by anything. So yeah, just respect them. They are wild. And then just a couple last tips. How do I get those photos? I waste a lot of storage space on my phone for folks who have a iPhone, or if you don't, an Android phone, you could do the video method as well to do screenshots. So with the iPhone, I use the live feature, and that means it saves like a clip of video, and I can scroll through that video to get to the shot I want to help with the ID. So again, that's how I get the different angles we're looking for insects, because even the experts, well, the entomologists may not even, with all the photos, might not be able to get down to species or even genus. So as many angles as we can provide is helpful to them. And like Jing-Yi said, there's observation fields. I also wanted to highlight one. This is the pollinator plant or plant pollinator interaction. You can do this with tags as well. If anyone's paying attention, I have different species on the screen, so I apologize. But if you're looking at the American bumblebee, you could put the American bumblebee, whatever plant it was feeding on, as a tag. Or if you look at my example, I actually had a mega chili, which is one of my favorite bees, on a butterfly milkweed. So you can tag butterfly milkweed, or the Latin name, in the tag. And then you can also add that as in the plant pollinator interaction. And like Jing-Yi showed, it'll save, and kind of it looks confusing on the back, like once it's saved into iNaturalist. But to someone's question about like, there was a question earlier that made me think about, why is this helpful? Do people actually use this data? There are researchers definitely using data now, and there's maybe a researcher who's coming up that might want to use this later. So the most we can do to provide the most useful observations for researchers coming down the line would be the ideal, I think, for a lot of us super users. Super quick, wanted to highlight another group of organisms that might not be on your radar to think about as having different angles. So I looked at my iNaturalist. I've only taken one picture of an earthworm. And I did not realize that. I've only taken one. I've seen tons of different worms, and including leeches and those sorts of things. So this is from 30 Legs' journal. It's photographing earthworms in iNaturalist. And I think the theme again is show the underside, very similar to what has already been said by Will and Jing-Yi. you know, if you don't get the other side, you oftentimes can't identify the species. And here's an example from that blog that shows, to my eye, it looks like a worm, but to the expert, this kind of shows you that there are a lot of different aspects of the anatomy that they're looking for to help them with identification. Just want to highlight some quick some other groups of organisms. Bats. We do have bats in the Chicago Wilderness Alliance region. I just, again, highlighting Chicago and Illinois. I know this map is wrong. This is why iNaturalist is not the only answer, but I know we have bats on the southwest side of the city, just not represented here. So we do have bats, and as you can see in the images, they're high in the trees or in the buildings, like at the Field Museum. We know that they're roosting not just in our trees, but in the top of the buildings. So there are a lot of different critters around. It's a very big world, even in cities like Chicago. Great diversity of, it's not just a lichen, or it's not just a spider, or it's not just a garter snake. There's a lot of diversity here, and we hope that we've opened your eyes a little bit to maybe keeping your eye out for things you weren't already documenting. And I'll pass it to Will.
All right, I'm going to zoom through some tips for getting good identifications once you've made your observations. iNaturalist does have a computer vision model, basically like an AI that is trained on all of the past observations that people have made and have been identified to research grade. And it's gotten pretty good. So like when you upload an observation, there's going to be, it's going to ask you for some kind of identification as your initial guess, even if you don't really know. And it'll provide suggestions for what it might be based on, the previous data that this model's been trained on. And so like usually these are pretty good if you have good photos. But if you have bad photos, then the suggestions that you get may not be good. There's also certain groups of organisms that are kind of underrepresented. And so, we always want to like ask ourselves a few critical questions when we're evaluating these suggestions that the model's giving us before we choose an ID from the list. I think you really always want to check the top several suggestions. It'll like give them to you ranked based on confidence, right? It'll have like a percentage score. And this is a little bit different depending on which app you're using, like which version of the iNaturalist app. But the new app has this like confidence score that I find pretty useful when you're trying to decide. So that's like the first thing that you should check. If it says like 99% confidence, then I would not feel bad about going with what the computer vision model suggested. But like here's an example of a mushroom that I found while I was doing field work in Peru last year that I did not get a correct identification for using the computer vision model. And you can see that like the top guess, the top ID suggestion is the genus Trichomopsis, but it's an 86% confidence, which is not terrible, but that's not like that good. And so, you want to just be a little bit skeptical if you see lower confidence scores like that. And then like just thinking about, does it pass your sniff test? So if we go to the next slide here, like some things that you should be asking yourself is like, is this the correct kingdom? Like, is this the correct type of organism? Something that I get a lot with like bad photos, especially, but even sometimes with good photos, if it just like doesn't recognize a particular organism, is like it'll tell me that a mushroom, it'll start giving me like suggested plants or something. And you can click on each suggestion. There's like a little eye for information that you can click on, and it'll take you to the page that we can see here on this slide, where it has some information about the species that it's suggesting, and it'll list the taxonomy there, like what kingdom it is, what phylum, family, etc. And if you can see that it's like totally in the wrong group, like if you're like, I'm pretty sure this is a fungus, and right now it's suggesting a kind of plant, then that's probably wrong, right? Another thing that's good to check is the range. So, on this taxon page for the species that is being suggested, it'll show you a map of previous observations of that species. And we can see here this Trichomopsis aurea, the mushroom that it was suggesting for my photo, If you look at that map, this would have been like the southernmost observation of that species ever recorded. That's not to say that it's impossible, but it's like it's unlikely, right? That this is going to be the right ID if I'm recording it significantly outside of its previously observed range. So those are just some things to check when you're trying to decide whether or not to trust an ID that the computer vision model provides. Another thing you can do is so when you get a suggestion like this from the computer vision model, for example, this Trigolomopsis aurea mushroom that it was suggesting as the top one, on the taxon page, and this is what it looks like in the website view, which, like, by the way, for a lot of these advanced kinds of features, you really want to be using the website. The app is great for like making observations, but like for actually looking at stuff and identifying stuff, I would definitely recommend using the website. And at the bottom of the taxon page on the right there, you can see there's a tab that's called similar species. And so if you click on that tab, it'll take us to a screen that looks like the next slide here, which is a bunch of other species that are commonly misidentified as this species. And so like if you're identifying something and you're not sure, it's always worth just like looking at this tab and seeing whether any of these photos that you see right here better resembles the thing that you've observed than what you thought it was. So that's just something quick that you can always do. And sometimes you're not going to be sure what it is, and you have to sort of just stop at family/genus, and that is totally fine. That's what you should be doing if you can't get it to species, right? The goal of iNaturalist is not to get every identification to species. It's to get every identification to like the maximum reasonable, knowable, taxonomic definition, which might only be genus or family, depending on how good the photo is, or how well studied a certain group is. Like there are some groups where the taxonomy is still really unclear, especially for like a lot of invertebrates and fungi and things like that. So in cases like that, one thing that you can do too is you can call in an expert, right? You can phone a friend. And so this one, for example, this is a Xylaria that I found in Peru that I could not identify the species. And it's actually, this might be a new species to science. So I'm sort of working on getting this one sequenced, the DNA sequenced, to see if that's true. But I happen to know that there's this guy, Fenner Morse, who is an expert in this genus Xylaria. And so if anybody's going to know it, it's him. And so what I did here, you can see in the comments of this observation, I tagged him. If you write @ and then you write their username, that's how you can tag somebody. So I asked him, hey, Fenner, what do you think this is? And if you don't happen to just know, who the experts on the group of interests that you're trying to identify is, a way that you can find that is by going to like the genus or the family that you're pretty sure this is a part of, and then looking. Every like genus or family will have a list of top identifiers of that group on the taxon page in iNaturalist, and so you can actually look at that list and then like call somebody in based on that list, which is pretty cool. Real quick, some advanced features. One thing to note is that observations have geoprivacy settings, and depending on what you're observing, you might not want the location to be public, right? So for some species, like endangered species, it's automatically going to be set to obscured. iNaturalist has this built in, which is great. But not every species whose populations might be in danger by human activity are automatically going to receive that treatment. So for example, if, I don't know, let's say you find some kind of orchid in a forest preserve, those are things that people like to poach. And so like obscuring that observation might be a good idea. Another thing we can do is we can create, or no worries, we can create or add to projects. This mostly is like not going to be necessary, but this is something that you can do if you have like a specific BioBlitz event, for example, that you're hosting, like we have as a part of City Nature Challenge, or if you want to like create a little challenge amongst friends, like certain individuals can join a project. So basically, there's like two ways that observations get added to a project. Either the user will join the project, or observations that match the project criteria will be uploaded to iNaturalist, and they'll automatically get counted. So for example, I could have like an Illinois mushrooms project, right? And I could set that project up so that every mushroom that somebody uploads from the state of Illinois automatically gets added. So that's one thing that projects can be useful for. But on the next slide, I'm going to show you why actually making a project for whatever specific set of observations you might be interested in might not actually be necessary. And the reason is that iNaturalist has an advanced search feature. And again, this is going to be much easier on the website than it is on the app. But actually, with the new app, you can kind of do this on mobile. And this, so if you go to like your home page or what's called the explore page of iNaturalist, you can see that tab in the sort of top left, explore. That'll take you to like the search view where you can search for observations. And there's like a little button that is sort of in blue on the middle of this like search square that I don't know if you can quite see, that says more filters. And if you click on that, it'll like expand additional search fields. And so basically this screen shows like all these different filters or parameters that you can search by. So you can search by like type of observation, wild, captive, things that are research grade versus things that need ID. You can search by a particular person. Like if I only want to see, bugs that Nicole is uploading, I can search just for observations by Nicole. You can search for different like groups of organisms. So there's these little icons right in the middle here. And right in this example, I've just selected fungi. You can search just for a particular place. So I've selected, I only want to see fungi from South America. You can even search by like date observed. So if it's like June and I'm interested in knowing what kinds of fungi might be growing in my area during that particular month, I can go over to the date observed and select only observations that were made in June of fungi in South America, for example. So this is a really powerful tool. And it's why projects are kind of unnecessary, because like if you are interested in a particular group of observations, you can probably just make a search from them or for that group, instead of like having to make a project for the Illinois mushrooms example. I could just search mushrooms observed in Illinois. There's no need to make a project for that. So anyway, this advanced search is pretty cool. Here's like an advanced thing you can do for search. This blew my mind when I first learned about it. So like every iNaturalist search, when you like are, on that previous screen and you're like putting in different filters or whatever, what that's going to do is that's going to create like a URL for that search, which is handy because you can like copy and paste after you've done your search and you see all the results that are there. You can copy and paste that and then, go back to it later if you want, if you save it somewhere. But you can also modify that URL with additional search filters that are not in that normal advanced search bar. And so one of the things you can do is search by observation fields. So here's an example that I use for my fungal work that has been really convenient for me, which is searching by the observation field voucher specimen taken. So sometimes I collect specimens when I have permits for them, and will submit them to herbaria or get their DNA sequenced. And I want to be able to keep track of which ones those are. So I fill in this observation field, voucher specimen taken. I click yes. And then it's saved in iNaturalist that I have saved a voucher specimen for that individual. And I can search for that observation field to get a list of all of the observations I've made that have that attribute. And so you can see the way that you do that is by adding an and then field, colon, and then the name of the observation field, except you have to put like % and 20s as spaces, which is a little bit annoying. But and then equals just means like if there's anything in there at the very end. But you could technically say like, yes or something like that if you wanted to be. I usually don't fill it in if the answer to voucher specimen taken is no. But if I was doing this field for all observations, including ones that I didn't take a specimen for, then I would want to set it to like voucher specimen taken equals yes in my search. So this is pretty useful, and you can do this for all observation fields. You can also use one of these advanced searches as like a jumping off point for identifying for other people. So I know a lot about South American mushrooms, and so sometimes I like to go and identify other people's observations. So what I can do is in the same search bar where I've set up my search to only show observations from South America that are of fungi, down in the bottom there's a little button that says identify. And if I click that button, it'll take me to the next screen here. And this is like a view of all of the observations that have been uploaded. And you can search and filter based on date and things like that. And what's neat about this is that it's a way to like very quickly go through lots of observations. It's like more efficient to click through this screen than it is to just, search and then click on observations one at a time. This like shows you a bunch of them, and you can very quickly like input an observation and then move on to the next one. And like when you've reviewed one, you can like click mark as reviewed, because sometimes there'll be one that you look at and you're like, wow, that's a bad observation. There's not enough information in this photo for me to be able to say what it is. Or maybe there'll be one that, you know, you look at and you're just like, yeah, I don't know what that is. Maybe it's identifiable, but I don't have the knowledge to do that. And so it's not going to be worth my time to see it again, you know, until I've learned more. So you can click mark as reviewed, and then it won't show it to you again, even though you didn't actually give it an identification. So yeah, those are some advanced tips. There's lots more that you could do. I feel like iNaturalist like advanced features is there's like so much more, and this is kind of just scratching the surface. But I'm gonna hand it over to Nicole at this point to just talk a little bit more about the City Nature Challenge in Chicago this year.
Yeah, thank you so much, everyone, for sticking around. I know we're literally at the end, just answering some questions about how to join the challenge. Laura has been putting the link in the chat. It's basically at chicagowilderness.alliance.org, or if you search in the iNat app and in the iNat browser version and projects, you can also find the 2026 challenge. But before I highlight that, I just want to point out, if you are curious to be, how to become obsessed with observations like the three of us are, there's a plethora of resources out there, YouTube, iNat itself. So please check those out. Here's some messy screenshots of the Chicago Wilderness Alliance webpage, but just wanted to highlight, if you want to join, if you are with an organization or you're a volunteer and you want to lead an event, that deadline is April 10th. There's a lot of Illinois representation. So I saw at least one Wisconsinite in the chat. So Wisconsin, we need some more representation. Where's my Northwestern Indiana folks at? Definitely need some more love here to represent all of our states in our region. But that deadline to add your event is April 10th. And then the City Nature Challenge itself is April 24th through the 27th. You do not have to attend one of these 2026 events to participate. Any documentation, any observation that is submitted, again, not cultivated, as we're saying, not that tree that's in your parkway. Any observation will be counted, regardless of if you're at an event. So the 24th through 27th, please make sure you're out and about, trying to document what you can. We're going to have, because I think I forgot to highlight that Will and I and Laura, who's our host today, we're all part of the Chicagoland City Nature Challenge organizing committee. And we're having at least two events. One is going to be in a brewery in the evening on May 6th, and one will be at the Field Museum morning of that Friday, the 8th. Those will be posted to the website soon. And then the last day to upload observations. Because again, if you're taking them on your camera as an example, and you need to go through that upload process, the last day to upload will be May 10th. Or if you're me, I need to make sure they move from my photo library to the iNat app. And here's another way to join. This is the iNat view of the project on the browser. You do not actually have to join this for your observation to be counted. It will be counted automatically. But if you do want to join to receive updates, where I have the red box in the top right-hand corner, it says leave in your screen, but if you are not part of the project, it will say join. So you can click that and you can join the project, and it's actually 17 days away. So we hope that you will join us, and we'll see you maybe at an ID event in May. Thank you, everybody.
Great job. Thank you so much, Nicole, Jamie, Will. Thank you for joining us. And like I said, this is recorded, and the PDF of the slides will be added to the website and sent out to everybody who registered. So thanks for joining us today. Are there any other questions? And I see that everyone stayed on. So if anybody has any questions, feel free to unmute yourself at this point.
But we can stay on if folks have questions. I'm sure Will or Jing-Yi, I don't know. I'd have questions. There was a questions. I know Laura who left already about gulls, Jing-Yi. They asked if they're all wasps.
Yeah, I think Will answered it.
Mm.
So it's not only wasps, but also mites, flies, and it also depends on how you define goals, because I think there's different definitions. So